really happy to introduce David Steensma from United States to give a talk about bridging the canyon between discovery and therapy. I think it's an excellent talk for the end of this meeting and leading up to the next meeting in 2021 in Toronto. So David, please. Thank you.
Thank you. That bridge is from Glen Canyon in the American Desert Southwest. So, colleagues who are still here, my friends, we have a problem, and it's a big problem. And it's not restricted to the field of myelodysplastic syndromes, and it's not restricted to the field of hematology. This is a problem faced by many areas of medicine. And it's not an insurmountable problem. I believe this is a problem that is fixable, but it is particularly pernicious. Maybe we should put a positive spin since it is a fixable problem and call it an opportunity or call it a challenge that we need to overcome. And I think our problem, our opportunity, our challenge is illustrated well by this amazing meeting, which has really grown so much over the nearly 20 years since I first came as a fellow. And when I was a fellow, I came and listened to rapt attention with people like John Bennett and Terry Hamblin and Eva Hellstrom, who were just names from papers, and they were real people and they were very human, and Terry was certainly very human indeed. We remember him. And over the last 3 days, we've heard about some terrific science related to MDS pathobiology, made possible by some tremendous technological advances. And we now, I think, understand this group of disorders much better than we did even 5 years ago. Imperfectly still, but we've made tremendous strides as an MDS community over the last 5 years. But I think everyone also recognizes that what's happening in the clinic has really lagged behind, way behind, years behind. And we'll talk about some of that. But first, a quick tour. I have nothing relevant to this topic to disclose other than my sadness about what's going on in the clinic. First, a quick tour through some of these, some of the coolest recent developments. I mean, it's now possible to do single-cell analysis at the DNA level, at the RNA level, increasingly at the protein levels. And no longer are we limited to analysis of homogenized bulk cells. This is a really remarkable technical achievement, and it's led to a better understanding of both normal biology and pathobiology. As a result of these discoveries, we've had to get rid of some sacred cows like this hierarchical hematopoiesis. We now understand that just like Darwin's intermediate forms, there are all sorts of intermediate states in hematopoiesis between what were once thought to be discrete species of cells. There have been tremendous advances in understanding epigenetics, and I very much enjoyed Peter Jones' talk the other day about how things have evolved over the decades. It's now possible, for instance, to do sequencing of single bases and look at their epigenetic state without destroying the sample, the way that we have done for many, many years with bisulfite modification. One very interesting example of such a library at the Broad is the repurposing hub, where they have hundreds of compounds that have previously achieved regulatory approval, so we know something about their safety. We know that they can go into human beings, and they are usable for testing and experiments to see if maybe they do something interesting that might be relevant to diseases like MDS. And we now know about dozens of molecular abnormalities associated with these diseases. When I started out in this field, we only knew about KRAS, NRAS mutations, 1988, RUNX1, and TP53 mutations, and that was it. ATRX mutations, rare but interesting, were something I was involved in very early. But 10 years ago, all of these most common mutations, DNMT3A, TET2, we didn't know about any of that. And I could go on talking about advances in the microenvironment and about immunobiology and so on, but I think you get the point. How many of you in the room take care of patients? Just raise of hands. So it's a lot of you. How many of you heard something over the last few days that's going to change what you do next week when you get into the clinic. Not in some theoretical day when we figure out how to manage the cytopenias with venetoclax, or when luspatercept gets approved, or APR 246. But I'm going back, I'm going to see patients on Monday, and am I going to do anything different based on that, what I've heard here over these last few days, even though the discussions have been very stimulating? And I'd have to say, no, I'm not going to do anything different. And that's because, by and large, when we sit with our patients, we're using drugs and we're using techniques that are years, even decades old. We're using prognostic scoring systems that are very helpful, but that are based on karyotyping, which really started in 1956 on the other side of the Oresund with Chio and Albert Levin. 1956, they figured out how many chromosomes we have, so hasn't changed much since the techniques that they used back then. Histopathology, it's very important. John, led the efforts to identify MDS as a discrete state. It wasn't just all acute leukemia, and that's been a critical development. That's part of why we're here today. But these are techniques that have been with us for a while, and that's still what we use as the foundation of how we risk stratify patients. And occasionally, we can even offer a cure. There's a few hundred people around the globe every year who get a stem cell transplant for MDS and are cured by. It should be many thousands. There are still people who think 62 is too old to get a transplant and never refer their patients to a center. This happens all too often. There are far too many people who don't have access, particularly outside the developed world. Their stem cell transplant is finally becoming a more global phenomenon, but it has been slow in coming. But still, I think we can celebrate this really as a partial success. But when it comes to drug development, we're really not in a very good place in MDS. So we have 3, now 4 FDA EMA-approved drugs. One of these, lenalidomide, is really a very minor chemical tweak of thalidomide, which was synthesized in 1954, the year Boeing had the 707, which is probably safer than the 787 MAX was, but so that the progress, the airline industry is often used as a model for us, and they keep saying, why in medicine aren't you guys more like the airlines and their safety programs? And maybe that we'll have to find another example. 1964, azacitidine was synthesized and was first used in the clinic a couple years later. That was also the year the Beatles first appeared in the US on the Ed Sullivan Show. The Beatles broke up 49 years ago. We have yet to break up with azacitidine. I wrote for azacitidine 8 times last week. Azacitidine is terrific. It's like rolling the dice. Sometimes you see these super responses. But we really need a Yoko Ono to kind of disrupt the whole thing. So this picture is not me, it's my mom and dad. In 1969, they got married. 1968, they were planning their wedding. We are celebrating their golden anniversary this year. That was also the same year decitabine was first used for AML. And I don't know if we should be celebrating a golden anniversary for decitabine or should be crying for its retirement. I don't know. And then finally, there's the most widely used class of drugs for MDS, ESAs, which don't have a regulatory label in the US. They finally do in Europe. And we shouldn't forget about them, even though since ESAs were first used in 1985, we've gone from the original Nintendo Entertainment System through 12 generations of consoles. And so this 8-bit stuff looks archaic. So about 10 years ago, a group of industrial process analysts based at Vanderbilt decided to look at clinical trials in the cooperative groups and what were all the steps required to get a trial open. And it took years for them to do this process analysis. And when they finally printed it all out on sheets of paper and brought it to the ECOG and CLGB meetings, it was the whole wall of a ballroom, almost this size, 8-point font. It was 10 meters long, 2.5 meters tall. And one of the most troubling things was they uncovered 70 steps that were actually infinite loops, where Committee A couldn't sign off on the trial until Committee B had approved it, and Committee B couldn't until Committee A had approved it, and it just got stuck at these various stages. They also found a number of steps that really added no value whatsoever, but were just there because some committee had power and had inserted themselves into the process. They called for a redesign. And I think in 2019, if we were to do this again, it'd probably be 12 meters long. It has not gotten any simpler. So you might say, okay, that's cooperative groups, and cooperative groups really, are a mess, and so institutions themselves are better, but that's not true either. They looked at Vanderbilt in particular, but I can reassure you the Farber is just as arcane. And there were also, whether it was an investigator-initiated or an industry trial, hundreds and hundreds of steps before it was activated. And when we collaborate among centers, every center has their own rules that need to be followed and enforces those. So this results in, and I'm not going to name the drug here because this is just meant to be a typical trial, but it is one that we opened at the Farber. Time from the concept, which was an exciting concept that people were enthusiastic about, till we finally got a full protocol, so that was almost 4 months. We got the full protocol. It took a little over almost 3 months to submit it to the IRB. Thankfully, we're now allowed to work on contracts and budgets in parallel to this, so that usually takes quite a long time. Development of the informed consent form, about which we'll talk more in a second, was the main hang-up here. So then got submitted to the IRB. It was scheduled 4 weeks later for a meeting. We weren't allowed to know what the IRB said until they sent us the minutes, which was another week later. It had some minor things that we had to respond to, which were exactly the same thing that MD Anderson had to respond to with their IRB, blah, blah, blah. And then once it was IRB approved and we had the green light, it was still 3 more months until we could consent a patient because we had to get more than 20 committees to sign off on it, including the radiation committee, even though the trial didn't have radiation, including the inpatient committees, even though this was an outpatient trial. We had to send, it had a patient-reported outcome with these little iPads like Reinhard talked about the other day. We had to send them to information technology so they could undergo a 30-day review to make sure they didn't have viruses that were going to bring down the whole Partners Healthcare and Harvard email system. And we had to build in our electronic medical record the templates for the drug. And so, that was another 91 days. We've had trials that this process has lasted as long as 300 days. We accrued, over the time the trial was open, 4 patients. None of them responded. That was just bad luck. And the contract research organization for those 4 patients generated 370 queries, and they're not done yet. I just got more while I was here at the meeting. Sorry, that we'll have to address. So I'm not going to bash CROs here, although they really have become a collective thorn in our flesh. I wrote this with Hagop and with one of our fellows, and I had originally called this Problems with CROs, but they said, make that challenges and opportunities. So that's what this is. And Hagop was a lot of fun to do this with, because he has a very dry sense of humor. And so, the CROs, they are important. They fill a niche. They are sort of what Uber and Lyft are, kind of, they fill the gig economy, so to speak, when a company needs to very rapidly expand its capacity and doesn't want to hire a whole bunch of people to do things themselves. And I get that, but part of the problem with CROs is they end up recruiting often people who have very limited, if any, clinical expertise, and in my experience, many of them don't have common sense either. And so this has become a real problem. And then, once the trial's open, the safety reporting has just become farcical. Simon Rule in Plymouth tweeted this consent form, and it's a real consent form. This drug could cause road traffic accidents, spider bite, food poisoning, or cuts. which is a little much. And I was thinking of, little Miss Muffet sat on a tuffet, and here she is reading her informed consent form instead of eating her curds away. And along came a spider and sat down beside her. And maybe because of the tibrutinib she was taking, she smelled better, it bit her, she had an accident on the way home. You can come up with why these might be drug-related, but it really is absurd. And, so I'm not going to keep picking on CROs. I've said my piece about that. But sometimes the trials themselves have problems, and we as investigators are to blame for some of this. Our pharmaceutical sponsors are certainly to blame for some of this. So we can do better. One thing we do as investigators, which we really need to be better about, is we always overestimate the number of patients we have who have a condition, who are going to be fit for a trial. We say, oh yeah, we see lots of patients with proliferative CMML, with SETBP1 mutations. And if we actually looked at the electronic record, we saw 2 in the previous 2 years. It's just those stick in our mind. And there's even a law, it's called Lasagna's Law. It has nothing to do with the Italian comfort food. It's Louis Lasagna, who is a pharmacologist, clinical trialist, and that is that investigators systematically overestimate the number of patients they see who will be eligible for a trial. That law was written in the 1970s, and it's still true today. We're also increasingly seeing trials coming from the solid tumor world into our MDS world, and this is welcome. We can very much learn from each other. But a lot of times the eligibility are just copied and pasted, and here's one that was recently sent to me, a proposal. Patients have to have a neutrophil count over 1,200, platelets over 75, hemoglobin over 9. In MDS, then what's the point? That patient probably doesn't need treatment, let alone this, which was for an HMA refractory group. There's nobody who's HMA refractory who met the entry criteria. So, and this sponsor was particularly stubborn about changing it. I mean, they finally did after investigators just pooled together, but it took a lot of effort. My colleague Andy Brunner, who's been a terrific addition to our Boston MDS community, is systematically looking at MDS clinical trials and their inclusion and exclusion criteria, and he's got a paper that he's preparing about this. But one of the interesting things is that these eligibility criteria are all over the map, and they have very little, if any, correlation to either the predicted toxicity or the mechanism of the drug. A lot of these include extramedullary disease, no evidence of blasts in the CNS. If you have blasts in the CNS, that's leukemia. It's not an MDS clinical trial at all. Uma Bharat at Oregon with Mikhail Sekaris and some others did an assessment of 140 trials to see what about therapy-related MDS, which is not an easy entity to define. People who have a history of chemotherapy or radiation may get MDS that is just coincidental and biologically de novo disease. And then we sometimes see these patients who look like they have therapy-related MDS from occupational exposures or some other thing. So it's not clean. But therapy-related MDS is about 10, 15, up to 20, depending on your series, %. She found it was less than 3% of trial participants. And rarely, I think, is therapy-related MDS actually a compelling reason to exclude patients. In fact, they do so poorly in general that it's a compelling reason to include them on clinical trials. And they were more likely to be excluded in industry-sponsored trials. Investigator-initiated trials, I think, we've all sat with these patients and realized how limited their options are. And so that, compels us to include them. Obviously, if they have an active other tumor that could make it difficult to monitor or interpret the toxicity profile, then that's different. So Andy made this lovely graphic, which I think summarizes many reasons why patients don't go on studies. This resulted from an analysis we had done in the MDS Clinical Research Consortium in the US, where we found that even at places like Dana-Farber and Johns Hopkins and Moffitt, big referral centers, only about 20% of patients ever went on a trial over the course of their disease. Nationally, it's about 1 to 2% based on the published data. And there are all sorts of reasons for that. It prompted me to go back. I see now between 200 and 300 new patients per year. I went back and looked at why the patients hadn't gone on trials of those who didn't, which was by almost all of them. And they either had a disease for which we didn't have a trial, they had a disease for which we did have a trial, but there weren't any slots, or they were from northern Maine or Pennsylvania, and they just couldn't come back and forth to the center, especially because they were elderly. And so there are lots of reasons why people don't go on studies. There's certainly a trend towards just adding stuff to azacitidine in MDS, and sometimes that's a great idea. We've seen some examples of preclinical work where there's a strong signal why something should work with azacitidine or why that would be a useful backbone. Right now, there are 50 different entities, I looked this up 2 nights ago, that are being combined in active or recruiting trials with azacitidine. Decitabine, there's a lot of overlap here. So aza has been the drug of choice for these combinations. And some of them are things for which there's compelling rationale. At least a few of them, and I'm not going to point to any here, it's because they failed as a monotherapy, and the hope is that maybe by combining with azacitidine, we can see a signal. So, we have a lot of challenges in clinical research. And, not to call anybody out here, but there have been numerous times in which, despite this, very senior administrators or scientists who don't see patients talk about, how much progress there's been against cancer and how easy it is to treat. One of the major pharmaceutical CEOs last October said, cancer is becoming ironically easy to treat. And I know it was a gaffe, and he meant to say that, the problem was with drug costs, which is a real issue. But, I just wanted to bring him to my clinic or help him meet the families of the 22-year-old that I had gone to the funeral the day before, who had therapy-related MDS from an osteosarcoma he had at 15. It just, when we have people like James Watson saying cancer is going to be cured in 2 years, or these Israeli scientists who claimed they were going to cure cancer in 1 year, which every patient asked about for the next 3 weeks. I know the MDS Foundation got lots of calls about this, everybody did. It really does make things more difficult for everybody, because there's a sense that, there's been this tremendous progress, and there has, and I don't mean to minimize that, but we still have a long way to go here. I think there's plenty of reason for optimism. I think these barriers are not insurmountable by any means. And there's a lot of encouraging signs. Number one, for MDS specifically, is there's been a huge uptick in funding. The Edward P. Evans Foundation has really built a whole community of researchers. There's other foundations. Dresner Foundation has been starting to fund some people. The Taub Foundation has taken an interest. We've long had support from LLS. We have MDS Foundation gives grants and so on. And so that's really been very helpful. And it's brought a number of young investigators with new ideas into our field and new approaches. And hopefully they're going to make the world a better place. There's continues to be good interaction between scientists and clinicians at meetings like this. AML, we went from 1991 to 2017 without a drug approval, a durable drug approval. And then there were 8 drugs approved in AML in a 2-year period. And I think we can do this in MDS. We're a little behind them, but I think we can get new drugs approved. Though all those clinical research problems I talk about are widely recognized, and there's been a push towards single IRBs. There's been a push towards tools to streamline that cooperative group trial so you can tell exactly where the trial is and where those infinite loops are and hopefully break the impasse. The NCI in particular and the EMA is doing this also, encouraging broader entry criteria, FDA also, to make a greater proportion of patients eligible for trials. I think, for those of you who see AML patients and are designing AML trials, it would be okay to include those patients with 10% blasts or more in those trials, even though they're MDS. I mean, they have a sort of similar prognosis. Low-risk MDS is a different kettle of fish. But I think we could definitely include these patients. I have to give a shout out for 3 different things that I think our colleagues at MD Anderson have done, which are really very nice. Number one, decentralized clinical trials. We have a trial going on right now in the Clinical Research Consortium in the US comparing different short schedules of decitabine, azacitidine, where patients can register at MD Anderson or Dana-Farber or at Moffitt, and then they get all the treatment in Vermont or in Georgia or wherever. This caused our IRB to have an absolute cow. And it took me a year to get through all of the, this, we're not going to have any control of this, blah, blah, blah. I said, no, that's okay. These drugs are FDA approved, and, we can follow how they're doing. And eventually we got it through, and this trial has finally met accrual. It did so very quickly because of this decentralized model. It doesn't work for every drug, obviously. Early phase ones, can't do that kind of thing. The second is inclusion of trials for people for whom trials are not available. So, Guillermo did this study with azacitidine, I think it was varinostat, or decitabine and varinostat. And you had to have a creatinine over 2 or an elevated bilirubin. You couldn't be eligible for other trials. I thought that was brilliant. I mean, it's not going to work again for every drug, but I thought that was great. And then they've also taken a new approach to contracting where instead of having to go back for each different combination of immunotherapies, they've gotten sort of carte blanche from companies to combine and do multiple different trials under a single IND and single contract. And so I have to give them credit for that. The MDS Foundation, we have to give credit also for helping lead patients to clinical trials. This is a big part of their website. And the publicity helps. Not every patient is going to, seek out a trial, but for those who do, this is a useful thing. Gail Robas, she taught me this, and it's actually worked. So, today, still, many of the CROs were saying, well, you can't enroll this patient because you don't have an FAB classification. The patient had a WHO characterization, and so Gail said, okay, we'll get you these FAB classifications, but it's going to cost you money. In fact, it's $5,000 just for us to even consider this request. And then every one where we have to do this is going to be an extra several $1,000s. And those requests just disappeared. And so, small victories. that was one. We are in an era where, despite this cooperation, we are seeing a rise in tribalism. Nowhere more so than in my own country, but it's going on everywhere. I am a tribe member as well. My patrilineal ancestor is Finn of Friesland. Sort of a few hundred kilometers away. The Frisians grew up in this sort of flooded land in the north part of what's now the Netherlands and into Germany and the south part of Denmark. And they were constantly at war with the Angles and the Jutes and the Saxons and the Geats who came across from Sweden. And Tolkien, who was in his day job at Oxford, an Anglo-Saxon scholar, did really the first analysis of one of the surviving manuscript fragments that dates from the same period as Beowulf, about this guy, Finn the Fries, who there apparently is both genetic and genealogic evidence that I'm descended from this medieval warrior. Finn even made his way into the Beowulf epic. In fact, in the 2007 version that had Angelina Jolie playing Grendel's mother, Finn came at the end of Beowulf's life, and Beowulf, said he essentially was already dead, but he killed Finn the Fries at that point. Partly legend. So this is how our ancestors treated each other. The hospitality of the Danes this week, and every time I have been in Denmark, has been tremendous. We are cooperating, even Denmark with Sweden are cooperating. I think we can build these bridges. We can work together. Even the Frisians and the Danes and the Swedes and the Geats can work together to make things better for our patients with MDS. So that's all I have to say.
Thank you for a very nice talk, David, and stimulating. My brain is a bit slow after yesterday evening, but it was really nice and really stimulating. I think, yeah, if there are any comments, I don't know, but otherwise, I think we will move on to the awards. And
Thank you.
But thank you very much, David.